joining us today. And we're pleased to have Jim McLaren here, a local Wellesley resident and a former high school teacher, to explain to us everything about genetics and what the information is that carries with DNA. So thank you, Jim. And this is part one, I should say, of the program. There's part two the following week on Thursday. So thank you.
Thank you. Well, welcome, and thank you for coming in on such a nice day. I hate to take you away from it, but I want to try to give you a sense this week and next week, if you decide to come back, of what's going on in the field of genetics. I try to give us some historical perspective as well, but what I'd like to do, just to get started off, is to talk to you a little bit about a tiny organism that has been used for most of a century to discover basic knowledge of DNA and how genes work. And it's called E. coli. It's a bacteria cell. It would take about 50 of them to stretch across a human hair. So we're talking about a pretty small little critter. I've magnified him here with a 2-liter Coke bottle because it's about the same shape, okay? Kind of cylindrical, rounded ends. And this would be magnified roughly 120,000 times normal size. This guy has DNA. He has genes that give him all of the characteristics that he has to live as a full-fledged living cell. He grows, he reproduces. I don't know why I'm calling it he, but and he's got DNA. His DNA, let me give you, I'm going to represent his DNA with this thread. This thread is actually about to scale in terms of its thickness to DNA. And I'm going to run off a length. Dave, would you hold this in for me? That is going to try to represent the E. coli chromosome, as it were. Tell me when to stop. Okay. Well, I'm going to stop myself because I'd be here a very long time. In fact, it just stopped for me. To give you a real serious idea, where'd the end go? It's this way. If I were to actually do the full length, it would be 3 football fields long.
Oh, wow.
Okay. And sort of weirdly, I gotta stretch it out here.
Oh, sorry.
That's all right. You were trying to be neat and clean, which is going to be part of the point. I'm going to tie the ends together. That's unusual. Bacteria do this. They like their DNA to be in a loop, a circular loop. So I'm going to tie it off here. There. Where did it all go? Here you go. Long end. There it is, okay? And this is only a tiny fraction of how long it would actually be, as I say. What was that, maybe 50, 60 feet? Should be 600 feet long in a loop. This guy, under ideal conditions in a laboratory, will reproduce every 20 minutes. He'll make a copy of that loop, pull one copy to one end, the other copy to this end, pinch off every 20 minutes. The next 20 minutes he'll grow, copy the DNA, pinch it off, and so on. Now, this was a bit of a simplification because I really should have run off 2 threads and spun them together, coiled them together about 500,000 times. That's what DNA is. It's a helix. It's this spiral, double-stranded molecule. Try to imagine this piece of thread separating, copying itself, and I will defy anybody in this room to take that out of here and just try to straighten it out. Okay? You've had enough trouble with the necklace in your jewelry drawer, right? And that's a foot long. Okay? So I just want to get a sense to you of what, every time I talk to you about molecules or, you know, this happens in this cell, it's it's beyond astonishing, okay? And let me give you some numbers, excuse me. This is a loop of DNA. It's a little tiny one. It's only about probably 10,000 units long. We're going to be talking about bases, A, C, G, T, bases. The subunits of DNA, this little loop is about 10,000 bases long. So just imagine 6 million bases long, and that's what we're dealing with here. This thing makes a copy of itself, okay? Here's the original loop, and the DNA has to unwind in order to copy, and so these two, one copy over here, one copy over here, and the cell in the confines of its walls has to keep them from getting tangled up. Okay. How fast does this go? 2,000 bases, units, A's, T's, C's, and G's, per second have to be copied in this 20-minute period of time. It has to rotate. It's a spiral, it's a helix. So in order to copy it, you have to unwind it. So it has to rotate at each of those points at 12,000 RPM. Okay? Your car engine would explode at 12,000 RPM. Okay? There are all kinds of mistakes. When you do something that fast, imagine typing 2,000 words a minute, okay? You're going to make mistakes, right? Well, mistakes get made. There are special editing enzymes that are charging along the molecule, finding mistakes and repairing them all along the way. And by the end of this process, that molecule will be almost perfectly copied. Might be 1 out of 6 million units, maybe one mistake that the enzymes miss, okay? It's kind of like you sitting at a typewriter, copying the Encyclopedia Britannica. You remember what that was? You remember what a typewriter was? You're sitting there typing as fast as you can type, and you make one mistake in that whole process. That's about the fidelity of the DNA copying process. So the mistakes are made, but they're very rare. That's a detail. At each of those copying points, the molecule is spinning to unwind. It spins in opposite directions. So down here, you get this tremendous torque accumulating, and it's starting to twist and bend and on top of itself. So the cell has to cut the DNA and relieve the stress and then pinch it back together. It's just an insane process, okay? And you got to keep those strands from getting tangled up in each other, okay? And how that is accomplished is simply a mystery to me. Always will be. And I will also mention at the time that this is a very simple cell. If you want to talk about one of your cells, which are reproducing all the time in your body, they're copying 1,000 times more DNA has to be copied every time one of your cells divides. So just multiply the insanity, okay, by 1,000, right? So just sort of keep that in mind as we proceed through our little discussion here. There's a name that might send shivers through your bones. Gregor Mendel, garden peas, high school biology, right? Tall plants, short plants, green peas, yellow peas, okay? If you don't remember that, it's because you've repressed it, okay? And so I'm not going to spend a lot of time talking about it. He's an amazing guy, and he did astonishing work, but that's a whole lecture that would put you to sleep instantly. He did his work in the 1860s, and he was totally ignored. He published his paper, nobody read it. Anybody who did read it said, no, that's wrong, can't be. And basically his work got buried. Rediscovered in 1900-ish. In the next 20 years, we call the golden age of genetics. Fruit flies were the primary organism used to discover how genes were inherited, how traits, visible traits. Fruit flies have red eyes, but every once in a while you find one with white eyes. And so you can cross the flies with each other, see what happens in the next generation, and so on. Hundreds and hundreds of traits in fruit flies and other organisms in other labs. But basically, the upshot is that Mendel's rules, very precise rules of how these things get passed, were expanded greatly, and Mendel's information, he didn't use the word gene, that didn't come about until the 1900s. But by 1920, we knew that genes were located on chromosomes, okay? So I need to remind you about chromosomes. Basically, they showed that chromosomes were essentially linked genes, one on top of the other in linear sequence on the chromosome. There's a picture of chromosomes. They're found in every cell nucleus in your body. This photo is plant cells, okay? About that distance is about the width of a human hair, okay? Just to give you a sense of size. When you look at cells, this is the outline of the cell, plant cell wall. This is the nucleus. And 99.9% of the time, this is what nuclei look like. They're not very interesting, okay? But every once in a while, the cell says, I've got to divide. I've got to make a copy of myself. And so the somewhat random appearance of dark staining stuff here starts to look organized and starts to begin to separate itself out into very distinct strands. These are chromosomes. And it took a long time to work out. What does that mean? Well, the cells are dividing and they're copying their genetic information. And we'll see some more examples. Every cell in your body has the same number of chromosomes, which in you is 46, right? 46. Okay? I say every cell, a couple of exceptions. But every cell has 46 chromosomes. When you and I were in high school, your textbook said 48, but that's because they're very hard to count. At least they used to be very hard to count. We now have methods that make it easy. And somebody said, whoops, it's 46. So, you have 46. If you're a fruit fly, you have 8. Okay? If you're a pea plant, you have 14. The number of chromosomes is specific to every species. It's pretty much got nothing to do with how complex you are. There are butterflies with over 200 chromosomes in each cell. There are some worms with 1. Okay, bacteria 1. So, but it's important that the numbers stay constant from one generation to the next, right? Because you don't want to bring an egg and a sperm cell together with 46 chromosomes each and get up 92, okay? Because that gets carried away after a very short period of time. So, the interesting thing is, people knew about chromosomes a long time ago, and they understood the behavior. Well, they occur in pairs, which is just like genes. Genes occur in pairs. Blue eye, brown eye. Okay? You have 2 genes in every one of your cells for eye color. Maybe blue, maybe brown, maybe both the same, one of each. Egg and sperm cells, this is the exception, only get one of each pair of chromosomes. And you only get one of each pair of genes as well. So chromosomes behave like genes. Fertilization brings the 23 back to 46 in that cell, which is going to become a new person, right? And the magic there, and I'll just mention this in passing, is that that cell has all the information, the 6 billion base pairs, that you need to create a human being, okay? When that cell divides, 46, those cells also will create a full human being. That's how you get identical twins, okay? I could take a liver cell from you, treat it correctly, and grow your identical twin from it. So every cell maintains all the information throughout its life. So let's get into some of the deep water here. Okay. By now, by the end of the 20s, we knew a lot about how genes express themselves, because we'd identified all these characteristics: red eye, white eye in fruit flies, blue eye, brown eye, human beings. Many organisms, they all followed the same rules, okay? And genotype means what genes did I pass from one fruit fly to the next. Phenotype, what did I look like? Did I have red eyes or white eyes? So the question became, by the end of the 20s, that's great, we know how they work, we know how they behave. How the heck do they work? How do they do that? How does a gene turn into a red eye on a fruit fly or blue eyes in you? And that sort of involved knowing what the genes were actually made of, and nobody knew. And then, even if you find out what it's made of, will that tell you how it works? Okay? How it does ultimately produce your eye color? These were totally wide open questions. Okay? Well, we know genes are on chromosomes. So what are they made of? Well, before we go any further, if you don't learn anything else today, understand that whatever a gene is, whatever it turns out to be, it must have two properties. It must carry information, okay? Somehow encode the instructions to make blue eyes. And it must be able to replicate. That means copy yourself. I should have just said duplicate. Biologists like to play word games. Replication refers to copying with the possibility of making mistakes. Okay? Duplication means put it on a Xerox, get an exact copy. Right? When you replicate 6 billion bases, you're going to make a few mistakes. Okay? So information copying. Information copying. Okay? Well, based on that, people knew that chromosomes were made of two things. They're made of protein. They'd known that back in the 1800s. They're made partly of protein. Oh yeah, and they got this stuff called nuclein that it was there too, but it's too simple structure. It was just totally ignored. Okay, protein had to be what genes were made of. They have this extraordinarily complex structure. Okay, this is a protein. It's called myosin. You don't need to know that. And it's a kind of model that we use. It's called a stick model, and all that means is that each of the sticks represents a bond between two atoms connected. And so here is on a little ring here, there's a carbon atom at every corner. and a carbon atom out here at the tips and so on. There's probably, I don't know, 5,000 atoms in this molecule, okay? It looks complicated. Let's just leave it at that for now, okay? And this is a fairly simple protein. And based on that, excuse me, the complex structure implies the possibility of carrying information, okay? It has a really complex shape, and maybe that shape tells the cell something. We like to use the example of a key. A key has information encoded in it, right? Because it will fit only one certain lock and open that lock. It has the information required to open the lock. Well, shapes like that, you know, why not? Entirely possible. They are absolutely ubiquitous. There is no living thing, there is no virus that doesn't have protein, okay? Proteins perform this insane number of functions in cells that are crucial. So they have so many functions, why maybe information carrying is one of them, okay? Could proteins copy themselves? Well, if you ask people that at the time, they'd say, I don't know, maybe, okay? I mean, this is just an example of the things that they do. There are thousands of proteins in every cell in your body. Some are structural proteins, some are contractile. The proteins in your muscle tissue actually grab onto each other and pull and move, okay? Hormones like insulin, it's a protein. Antibodies are proteins. This group down here, enzymes, okay? These speed up chemical reactions. There are thousands of enzymes in every cell. So they just have this crazy number of functions, so why not being a gene among them? Well, I'm good. Hang in there, guys. We're going to do some chemistry. Hate to do it to you, but it's really good to really understand how DNA works. You got to know something about proteins. So here's a quick and dirty lesson in protein structure. You've all probably heard about amino acids, right? Amino acids are relatively small molecules. They consist of we call it a central carbon, and carbon usually has 4 things attached to it. This thing is an acid group. It has properties of an acid. This side, called the amino group, nitrogen, a couple of hydrogens, has properties of a base, okay? And the 4th bond has something else here, some other atom or group of atoms on this 4th bond. So there are many different amino acids, all of which have this structure in common, and then something sticking off there. And all proteins are polymers. Polymers are just big molecules made by taking small molecules and linking them together into long chains. Simple as that. And that's what a protein is. That R group just means it's something different in each amino acid, every amino acid, and I'll show you a couple of examples. All proteins in all living things are made from the same 20 amino acids, okay? There are lots more than 20 amino acids. You can put anything you want right there Yeah, I don't know why I have a laser printer. Anyway, but proteins only use 20 of them. Okay, there are hundreds of amino acids. Proteins in you, proteins in oak trees, proteins in earthworms, proteins in bacteria, every living thing. The proteins in every living thing use just those 20 amino acids. That says something interesting about common ancestor, maybe, evolution, maybe, okay? But that's another story altogether. And here are just 6 of the 20, just to show you that they all have this same thing here, 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 here. But you can stick something else on that 4th bond, and each one has different properties, okay? So each amino acid, some of them love to dissolve in water, others hate water. Some of them have a negative charge, electric charge on them. Some have a positive charge, so they'll attract or repel each other based on that. So you don't have to remember any of that, just get the sense. So if I'm going to make a protein, all I got to do is stick them together. And here is the nitrogen, the amino acid, amino acid, amino acid, amino acid. You use that to form a backbone, and then that other thing sticks off of each amino acid. And so you can imagine if, say, these 2 guys hate water, they'll try to bend around. It's very flexible, and they'll bend around to get together to exclude water from the area. Or if they're both negative charge, they'll repel each other. And so what's shown as a straight chain here, will bend and twist and turn and get all knotted up in 3 dimensions. And that's the key part of proteins. And I'm not even sure I'll put this in here, but so a protein has 1 amino acid attached to the next one, to the next one, the next one, the next one, in these long chains. And the chain folds itself based on attractions and repulsions of those site groups. So you get a 3-dimensional structure, ultimately, emerges if you put the amino acids together in the correct sequence. Okay? And that's the key. You got 20 different amino acids. Think about a language. 20 different alphabets, or 20 different letters in an alphabet, right? And this is what can result in that twisting and folding that takes place. But if you put lysine next to alanine, next to tryptophan, next to another lysine, and then if you put them in that specific order, they will fold into the same 3D shape. Okay, that's the key. So go back for a sec. So the sequence of the amino acids determines this. You have the same sequence, you will get this shape. And the shape will determine the function, whether it's an enzyme or a structural protein or an antibody or whatever it is. Sequence determines 3D shape, determines function. So let's go back here and look a little bit at this molecules and information concept. So we're going to put together strings of 20 different amino acids, right? Well, English language is based on strings of 26 letters, right? You put them together in the right sequence and you get a word, okay? You get an F-O-R-M together, says form. That's a word we all recognize, okay? QXPZT is a sequence that is nonsense, right? So alphabets allow complex languages, okay? So 20 letters, a 20-letter alphabet would be perfectly capable of making a very complex language. I point out Hawaiian, okay? 12 letters in the Hawaiian alphabet. And they are Hawaiians are very capable of getting across any idea that we are or anybody else is on the planet, right? So, you know, what's the big deal? I'm gonna, later on I'll probably repeat myself, but biologists got really hung up on this. When they looked at DNA, they knew that DNA had an alphabet of 4 letters. They said, how can you make an alphabet, how can you make a language out of 4 letters? That's impossible. Well, if they'd gone back into the 19th century and asked any mathematician worth his salt, they would have said, I only need 2, thank you, 0 and 1. And I can express the most complex ideas you would ever want to express. So I think I've got that elsewhere, but I just need to make the point that DNA was ignored because it was so simple, okay? 4 letters is quite adequate, thank you very much. So a gene must not only encode information, which protein could do because it's got sequencing of amino acids, but it also has to copy itself. And there's no evidence that any protein can do that. But that may be just because we haven't found the evidence, right? So, a very short version. 1860s, Frederick Miescher discovers nuclein. He separated sperm cells and he discovered protein, and this strange stuff he called nuclein, which turns out to be DNA. He published the paper, everybody said that's nice. Now I'll go back to whatever I was doing. It was pretty much ignored. In 28, Frederick Griffith, Nobel Prize, discovers that he can transfer a genetic trait to a bacteria cell by exposing it to the contents of dead cells, other dead cells. And so it sort of planted the idea that, well, what did you take out of the dead cell to implant in the bacteria? And Griffith didn't know, and nobody knew how to figure it out until these 3 guys. They essentially proved in 1944 that it was DNA, that useless stuff that nobody knew or cared about. That was the stuff that transformed Griffith's bacteria. And all hell breaks loose, okay? As soon as DNA is identified as the genetic substance, every lab on the planet said, oh my God, what is this stuff? how can I figure out how it does what it does? And of course, the winners in that race were Watson and Crick, 1953, announcing the double helix. And it was really kind of, this was sort of the feeling, you know, DNA, you know, 4 letters. The structure of DNA, the components, DNA was known to have these 4 components, A, C, T, G, okay? And Watson and Crick, Wilkins and Franklin, using techniques that I won't bother you with, fascinating stuff, but they were able to crank out this 3D model based on data from these two to announce the double helix structure of DNA. Watson, Crick, and Wilkins are given the Nobel Prize. Rosalind is not included, partly because she's dead, and you don't give Nobel Prizes to anyone who's dead, but also not entirely because she was a woman. Okay, there's some real stuff going on there that you could read about. James Watson is not a nice man. I'm sorry, it's my opinion. Okay, but this was the announcement. It's a double helix. Okay, this is a nice picture. This kind of model. This is not sticks and so on. This is called space filling. Okay, so you can see the position of every atom. Atoms are not red and blue and green and white. Okay, that's for us to help visualize. So what's a helix? A helix is a spring, right? And a double helix means you take 2 springs and kind of cram them together. It's that simple, right? But with the addition of taking cross pieces and linking the 2 springs, okay? And that's where the interesting stuff starts to happen. I'm throwing this one in. This is, I don't know, 15 years ago maybe, development of a very new kind of microscope that literally visualizes individual atoms. And there's DNA, son of a gun, it's a double helix, okay? You can see it directly. So, just a quick primer. Like protein, it's a polymer. It's a long molecule made of individual subunits linked together, okay? Really long, as I showed you with the E. coli, right? You remember pop beads? There's a little, you know, about half-inch, I don't know, quarter-inch diameter beads. And you could pop them into each other. Think about proteins and polymers, DNA. Think about it that way, okay? And for proteins, you'd have 20 different colors, okay? Pop beads, string them together. For DNA, you'd only have 4 colors, and you could put them together in any sequence you want. Sequence equals language equals information. So you just have to imagine your necklace being 300 miles long.
But you need a double helix on that.
Well, yes, for DNA, you have to have 2 strings of pop beads, right? And and good point, and I'll come back to that in a sec.
I corrected the teacher.
Oh, what's that?
I corrected the teacher.
Darn right. You should correct the teacher.
Good to know.
Any teacher who doesn't like being corrected doesn't belong up there. Now, with pop beads, to make a typical DNA molecule, you'd have to do about 50 million, okay, to get that sort of an average length, and it would be about 300 miles long. Okay, so just again to give you a sense of scale here. And you would need 2 strands, okay? And and you'd have 4 colors, and the way you pair the colors is part of the trick. So here they are. Oh no, molecules again. Hate molecules. The only thing you're going to have to remember is A, G, T, C. Okay? This is what they look like. All 4 of these have this same common gizmo here. This is called a sugar. It's called deoxyribose. Deoxyribonucleic acid, right? And then there's this thing, okay? They're called bases because they have the properties of bases. And they're not identical. Even these two. See the 2 rings. But here there's a nitrogen there and an oxygen in that spot here. So they're very similar, but definitely not identical. Same of these 2. Okay, just a single ring, but you know, this up here on that's different, that's different. Okay?
Jim,
That turns out to be
how did they
important,
get these names?
but I'm sorry?
How did they get these names?
Goes back
For the 4 items, A,
That's a very good question. I have no idea.
goes back to the Greek.
Think about that one. It's when you take organic chemistry, you just memorize stuff. You know? You don't ask questions like that. So, DNA is a long chain made of simple subunits. Okay, these are the subunits. There are only 4: A, C, T, G. Okay, so keep that in mind. Okay, so the key to remember is A fits T, G fits C. They literally, when you bring them together, you make a model of the molecules, you bring them together and it's almost like this. Adenines and thymines almost do this.
They interlock.
But an adenine and a guanine,
Won't.
okay, they don't fit. C's and G's fit.
Interlock.
So, here's what DNA looks like. 2 strands is sugar phosphate backbone. That's the ribose, deoxyribose sugar, okay, and a phosphorus thing. That's why DNA is an acid, even though it's got bases in here. It's an acid because phosphate is a very strong acid. Don't worry about that. But you get the sugar, the phosphate, the sugar, the phosphate, the sugar, the phosphate, and so on. And on each one of those is one of those 4 bases pointing out. The other strand points opposite, and the A's and the T's fit together on either side, either way. And the C's and the G's fit together. So all of a sudden, and then it's kind of like a ladder, right? And you take the 2 ends of the ladder and you do this, you twist it, and there you go with your double helix, okay? So check this out, okay? A, C, G, G, T, T, C, G. That's information. That's words, okay? I don't understand the words, okay? But it's a language. The cell understands the words. So DNA carries information. No question about it. Okay? When Watson and Crick published 2-page paper, and it had one photograph and one diagram, so it was a very short paper. And it was purposely short because they were in a race. They knew other labs were working on this, and they didn't want to lose. So they bang, bang, bang overnight, 2 pages into print it goes. And this sentence is right near the end. And this is your CYA statement. This was cover your tail. It has not escaped our notice that the specific pairing we have postulated, A's with T's, C's with G's, immediately suggests a possible copying mechanism for the genetic material. See, protein can't make copies of itself. But the structure of DNA, just look at it and you say, oh my God, it can make a copy of itself. Okay? It's staring you right there in the face. How we doing here? Yikes. Sneaking up on time here. So here's a highly oversimplified but view of what's going on. To copy it, you got to separate it, the 2 strands. You got to pull them apart. The ATCGs are held together by weak attractive forces. So it's not that hard to pull them apart. And you get them exposed like this. Then you bring individual nucleotides, subunits, come up. They find partners, okay? They find the correct place to be. But they're just kind of floating around. It's a weak attraction. So somebody has to come along and bind them together, okay? And by the time you've done that, look over here, these are identical to each other. G, A, G, A, T, G, okay? G, A. These 2, same information that was here, is now doubled. Okay? Now I can send one of those copies to one cell, the other copy to the other cell. We've got the same information. Question?
So you've given us a particular order of the different letters. Now, would that particular order go off and become an ear? And if you change the order, go off and become a fingernail?
Yeah. That's a bit of a simplification, but yes. Okay? If you start messing around with this order, you are going to create problems, most likely. One little detail, I'll try to wrap up fairly quickly here. Here's a sequence of, here's a DNA sequence, right? A, C, G, C. Presumably the cell says, oh, I can read that. That makes perfectly good sense. Okay? And you say, well, you got this sequence over there because of the pairing rules. What about this? Does this say anything? And the answer is no. Basically, it's gibberish. And I give you this example. I'm going to make a DNA molecule out of the English alphabet. And I'm going to use a pairing rule. Okay? This strand is going to be the next letter in the alphabet. Right? So STU, right? HIEFCD, right? And you look at that and you say, huh? Okay? So why does DNA even have this strand? So it can copy itself, okay? And maintain meaning in the other strand. So we say that DNA has one strand is sense, makes sense to the cell, and the other is nonsense. And you have to ask the question, how does the cell know which side to read? Okay, that's a good question. Might get to answer it next week, we'll see. So sequence is everything, sequence is language, okay? And now, so now we have two languages. We have DNA language, 4 letters in the alphabet. We have protein language, 20 letters in the alphabet. It's like, you know, English and Norwegian, okay? Pick 2 languages. Somewhere, somehow, translation has to happen, okay? You gotta find some way, somebody who speaks them both and can go back and forth, okay? So where does that happen? Because it turns out, if it, in protein language, if it's the sequence of amino acid that determines the shape, and shape that determines function, then DNA is what must carry that sequence information that says put a glycine in the first spot and an alanine in the second spot and this amino acid in the third. And that, once Watson and Crick had sort of finished, that was the giant puzzle piece to be solved. How does it get translated? And very, very quick and dirty here, and then I'll wrap it up. Here's DNA language up here, okay? 2 strands. We're only going to read one of them, okay? It's called the template strand, is the one that makes sense. And to begin the translation process, we get this guy involved, RNA, ribonucleic acid. You've probably been reading a lot about RNA. If you've had your COVID shot, okay, you've been taking RNA. But RNA can make a copy along the strand of DNA that makes sense. And it uses the same pairing rules, except RNA is a little different, chemically different. It uses something called uracil instead of thymine, but uracil pairs with adenine just the same as thymine does. It's a complication, I'm sorry, it's not my fault. Life is not simple. But this RNA comes, reads the appropriate stretch of DNA, and then peels off, and it goes out into the cell and finds the place where proteins are made. And it says, this sequence, AUG, means put a methionine in the first position of a protein. AUC says put isoleucine in the second position. UCG says put serine in the third, and so on, and so on, and so on. This protein with this sequence will fold into a 3-dimensional shape determined by that sequence. So that's basically what's going on. Next week I'll try to extend this a little further, and then we'll get to the good stuff, okay, I promise. Because you know about genetic engineering and cloning and on and on and on and on it goes, and it's in the newspaper every day. And what I'm trying to do is give you enough gut feeling so that when we really talk about those methods, it'll, I hope, make some sense. So I think I'll wrap it up at that and take questions if you want, or sneak out the back and never come back, whatever. Yes, Roy.
This may be a silly question, but I was just thinking, okay, you've got the twist in it. But then I found myself thinking, well, why does it have to twist? Why couldn't it just be a parallel like a long railroad track?
Yeah.
Track?
It's because chemical bonds have restrictions between atoms of mobility. And the only way that you can get those bases next to each other so that they fit is because the sugars and the phosphate that are holding them have to follow that path. If you straightened it out, the bases couldn't get near each other.
And I'll be darned if I could get that straight enough to tell somebody else.
I know, I know that feeling, yeah.
But there's a good reason for it.
There is a good, very good reason for it. Yes?
Jim, I wish I had you as a high school teacher when I was a lot younger.
Oh, no, you don't.
When you started the lecture, you were talking about how cells were replicating each other so rapidly and so forth.
Yeah.
Are they also dying at that same rate?
Oh, yeah, absolutely, yes.
And what happens to
I
the
could,
waste?
I don't remember the numbers, but you are producing millions of red blood cells every second, okay? Well, that's because millions of red blood cells are dying every second, okay? And you have mechanisms to adjust that rate. You move to Denver tomorrow, and suddenly you will start making more red cells than die, okay? Because of the altitude, until you reach the right level of red cells at that altitude, and then you adjust.
So if you move to a higher altitude, you would be anemic?
You have trouble getting oxygen to your cells. You know, I've always wondered about the NFL saying, we don't mind going to Denver. The heck you don't mind going to Denver. You're 5,000 feet and you're going to be gasping for air, you know. Meanwhile, the Broncos are doing just fine, thank you very much, okay?
Played one hell of a game against
They claim
the
it's,
Giants.
no, that's no, yeah,
But don't you produce as much, you produce much more red cells?
Roy, yeah.
Just a quick question. I think I read someplace, Jim,
Weeks. Weeks.
every maybe year, we have completely
Replicate your body, is what he's trying to say.
replicated ourselves,
Yeah,
all the cells, by something like over 80%, 90%, something like that.
something like that.
Is that
Mhm.
right?
Yeah,
Okay, that's
Yeah.
what I thought that just boggles my
Oh, that-
mind.
I think about that stuff all the time. I mean,
I'm
you,
glad I'm not the only one.
there are parts of your body, not
Yeah.
parts of your body, there are atoms in your
Yeah.
body
Yeah,
that were in Julius Caesar's body,
yeah, yeah,
okay?
yeah. Dinosaurs.
Or any other
Yeah.
human being you want
Yeah,
to, yeah.
I'm still a dinosaur.
Yeah, we're all dinosaurs. Yeah.
But there are certain atoms, molecules rather, that don't really replicate, aren't there? Just a few that.
Oh, there are
that's
cells.
Not 80, or cells, yeah.
Neurons do not
That's what.
replicate.
That's what I couldn't.
They,
Yeah. Thank you.
you've got,
Yeah.
well, fewer. You have a certain number of neurons
Uh-huh,
when you're 18 months,
uh-huh.
and that's it.
Okay,
And it's only downhill from there.
just one deck of cards.
They,
Yep.
they will
Okay.
die, they will not
Yeah.
replace themselves, yeah.
That's a happy
Yeah.
note.
So, but your skin cells, cancer patients who take chemotherapy, chemotherapies are designed to kill dividing cells.
Yeah.
Well, hair follicle cells divide all the time and rapidly. Your hair falls out, okay? Digestive problems because your digestive cells lining your digestion replicate very frequently. So that's a problem. And it's a real tightrope walk, giving the dose that will kill the cancer cells and not too many of your normal cells. So,
Well, thank you.
yes.
I think it was the summer of '62. I had a temporary job as a typist in Watson's lab.
Oh, really?
I never met him. I had learned the skill of technical typing, so it
Yeah.
was a good part-time job. I was working for Manpower. And I said to the office manager woman, I said, it seems strange. I never met Watson. All the technicians busy in the lab are women. And she said, that's because if he had men, they would want credit on the papers.
He
was not a nice man.
he remains to this day not a nice man. Read The Double Helix was his first book, and this sexism is just dripping. His comments about Rosalind Franklin, he's tried to back up since then some, but
Is single man?
yeah, I think he is. He wrote a book 10, 12 years ago. It's called Avoid Boring People. And the double entendre of that title tells you a lot about James Watson. Anyway, yes.
Thank you.
So in 1953, what did the two of them have? What diagnostic methods did they have that they weren't available before? Was it imaging? Was it chemistry?
Yeah, they had.
Besides the mind.
They had the ability to do these models. They stick and that was Linus Pauling had sort of come up with that. That carbon has bonds that can only go in certain directions, and so molecules can only have certain shapes. But mostly, they had something called X-ray crystallography, which is I understand how it works. I can't even begin to describe. I'd show you the picture that they published that says, "Oh, it's a helix," when they first saw it. And I look at that and I don't, what? But that's the methodology. It had been around for some years, but it was hard to apply to DNA because it needs crystals. It needs solid crystals to work its best. And you can't crystallize DNA, but you can draw it out into long strands, and that puts enough order into the molecule that they got the clue they needed. But
That's how we saw the sequence.
yeah.
When did the electron microscope come out?
Electron microscopes have been around since the 40s.
Well, they used that.
Wouldn't the picture I showed you was only possible 10, 12 years ago because the improvements in the microscopes themselves. Electron microscopy really doesn't get you very far in that regard. It's a marvelous technology. Yeah.
I'm just curious about where the resources come from to conduct these kinds of studies. Like, for example, right now we're seeing all kinds of public and private funds drying up. What kind of resources do these people have?
Oh, good question. You go back far enough. You go back to Darwin. Darwin's resources were he married a Wedgwood, so he didn't have to work for a living, and that was true through most of the 19th century. By the 20th century, universities and labs, but it was really after the war with the GI Bill and the Manhattan Project, and the government seeing, ooh, it pays dividends to invest in research, okay? And until a year ago,
What's the name of it?
so it's a good question. And,
Projects are private and feds.
oh, sorry?
All those projects are either privately endowed or fed. The fed
Yes,
Has to be.
right. And I think we're gonna see a brain drain, you know, in the other direction.
Yeah.
Top 25 scientists in EPA have already been gone.
Yeah, big time. Well, thank you all.
Yeah, very good.
Appreciate it. See you next week if you can stand it.
Yeah, you're pretty good.